until the 60s that the neurons in contrast to other cells in the body are not reproducible, and this is a so-called stable population. Okay, and but it has been demonstrated that there are so-called stem cells in the organism, and stem cells they can give rise to neurons in certain regions of the brain. Do you know probably what are the regions of the location of in the brain of the location of stem cells? Which area?
Ventricles?
Well, so near lateral ventricles, in the hippocampus and in the striatum. Okay, and actually there is a number of other brain regions which are considered as candidates to produce, to collect, to keep stem cells. One of them is hypothalamus, but still there are no strong evidence that in the hypothalamus there is also a location of stem cells. Also, I will repeat once again that the idea of stem cells, it was generated by someone Russian. Do you remember his name? It was, he declared, he suggested that he expressed this idea of brain cells in 1908. Do you remember his name? That was Maximov. Maximov, he was a professor of the St. Petersburg Military Medical Academy, but at that time he immigrated to the United States. Okay, so Maximov. But the strong evidence of the existence of stem cells, they had been provided by someone Russian, Friedenstein, and I also mentioned that he was a son-in-law of Luria. You remember that? Well, and well, so he proved. But the idea of the stem cells, or the existence of stem cells in the brain, that was declared by many, but by a number of scientists, including Kern, and also that's very interesting story that my first supervisor in neuroendocrinology, that was a corresponding member of the Russian Academy of Sciences in St. Petersburg. Well, so he had all the evidence in his hands on the existence of stem cells in the brain, but he gave an incorrect interpretation of this evidence, of this data, and that was very pity. Well, so the stem cells are generated during the whole life. What are the properties, the characteristics? Well, so the brainstem, which are located in the brain in adulthood, they have two kinds of polypotency. They can give rise to neurons, and they give rise to glial cells. Do you remember what kind of glial cells are there? There are three types of glial cells.
Astrocytes.
Sorry?
Cyst-like cells. Cyst-like cells.
What kind of?
Okay, maybe.
Well, so there are three types, huh? Astrocytes,
Dendrocytes.
and
Microglia.
Okay, good. Well, and it was declared that stem cells proliferate, and they are realized in the genetic program under the control by extracellular signals. What is very interesting, that it has been demonstrated over recent years, over recent decades, 2-3 decades, that at all stages of the functioning of the existence of stem cells, they're under the control by extracellular signals, by hormones, by neurotransmitters, etc. Well, so the most important point in their fate, this is the arresting of proliferation of mitotic divisions. Okay, well, so after the arresting of mitotic division, they become undifferentiated or lowly differentiated neurons, right? Or glial cells. Okay, no neurons, sort of. Okay, because the glial cells they proliferate extensively. Good. Well, let's go on that. And now, what I mentioned that we could differentiate two types of neuroplasticity, at the cellular level and at the molecular level. And now let's turn to neuroplasticity at the cell level. Well, sorry. So first, it has been demonstrated, repeatedly demonstrated, that if you cut an axon of the neuron, sometimes it provokes the retrograde degeneration of the neuron completely, retrograde degeneration. But sometimes this axon can be regenerated. What does it mean? That it continues to grow, giving rise to a lot of axonal terminals. Axonal terminals. Well, so one of the metacellular mechanism of neuroplasticity, this is arborization of the lesioned axons with the appearance of numerous axonal terminals. Okay, this is the first mechanism. The next mechanism, and this is the second mechanism, that you know that, by the way, what are the compartments of the neuron? There are three compartments.
The body, the axons, and
Cell body, axon,
and dendrites.
and the dendrites. Good. Well, so there is a mechanism of the neuroplasticity which is realized at the level of the dendrites. Well, so look, this is an axon with axonal terminals. This is the target neurons. This is cell body, and these are dendrites. Okay. Well, so at the dendrites they give rise to so-called spines. Usually, spines they correspond to synapses and to direct contact with the pre-terminal axon. Well, so spines usually they are in tandem with pre-terminal axons. They give rise to if the number of synapses and therefore the number of spines which are involved in the construction of the synapse, it can be increased or diminished under different functional states of the brain, and that after the lesion of the brain also. And then you can see these are spines, this is dendrite, and these are axons, pre-terminal, presynaptic terminals. Look, what can be the difference in the course of neuroplasticity? First, there might be a small region, surface, of the synapse, a small region of the synapse between the spine and the presynaptic terminal, with only one active zone. What does it mean, an active zone? An active zone, this is a zone which is characterized by the accumulation of synaptic vesicles, which contain synaptic vesicles, they contain what?
Neurotransmitters?
Neurotransmitters, exactly. Well, so with an accumulation of neurotransmitters and the high density membranes, mostly postsynaptic membrane. Well, so at this region, there is a small region of this synaptic content. Okay, then if the neurons they function more intensely, they can be in a large area of the synapses, and in this case there are two or even more active zones. Okay, there are two accumulations of synaptic vesicles with two local regions of the dense membrane. Good.
So, painted red in picture, is this an axon or a dendrite?
This is dendrite, and this is axon.
Okay,
Okay.
so.
Well, so this is a synapse between pre-terminal axonal swelling, okay, varicosity, and the spine of the dendrite. Spine, very synaptic axon. Good.
Thank you.
Well, so, and the third mechanism of the intensification of the synaptic neurotransmission and the neuroplasticity, this is an increase of the number of small spines which give rise to synaptic contacts with the same axonal, good morning, axonal synaptic pre-terminal. Okay, good. Well, so there are three mechanisms at the level of the axons which are formed by the spines of dendrites and by the presynaptic axonal terminals.
I'm sorry. Can I ask a question? So in the lower right corner,
Yeah.
Does this mean that two spines can connect to one axon, or did I not understand correctly?
They can, the spines what?
There are two spines there.
Yes,
So two spines can connect to one axon?
yes, yes. Yeah, not even to one axon, to one axonal varicosity, axonal swelling, as it is drawn here. Understand? Yeah.
Yes.
Well, so there is only one axonal terminal with only one spine. There are one axonal terminal with one spine, but with two active zones. And there is one axonal terminal or varicosity with two spines with two active zones.
Can there be more than two?
Sorry?
More than two can there be, or is this maximum?
Not maximum, not maximum, but this is very typical case. Yeah.
As far as I understood, there are two.
This.
The two dendrites are on the same the one post synaptic neuron, right?
Yes,
Is there any-
these are postsynaptic region of the synapse.
Yeah, but is there any option that to the one axon can be connected several different dendrites,
Yes.
different neurons?
Yes, that can be. That can be also. Well, so these are, in fact, these are so-called cellular mechanisms of neuroplasticity. Okay, and now let's turn to molecular mechanism of neuroplasticity. Well, so according to the dogmas in neurobiology of the beginning of the previous century, each neurons can synthesize only one neurotransmitter. Okay, but in fact, after the development of different techniques like multi-labeling of different antigens or different neurotransmitters, it has been discovered that that the neurons, individual neurons, single neurons, can synthesize several different neurotransmitters. And somebody put me a question during our previous lecture, I think. Well, so there are the examples of neurons which produce several neurotransmitters. Then, if according to the same dogmas, the neurons should have a very conservative genetic program. It means, and they should be very conservative. And therefore, the synthesis of neurotransmitters are very conservative processes and the expression of enzyme of specific enzymes for their synthesis are very conservative processes, and they cannot be changed over life and in different functional states. But in fact, expression genes and synthesis of neurotransmitters or enzymes of this are regulated by intercellular signals. Well, so if there is a change in the intercellular composition of chemical signals like hormones, like neurotransmitters, okay, well, so it could influence, they could stimulate or prohibit the expression of specific genes in the neurons, which are involved in the production of neurotransmitters. It's clear? Not really? Clear? Okay, good. There's no number. Then, the second one. Neurotransmitters affect receptors not only in synapses, but also along the whole neural surface, providing the volume neurotransmission. Also, we have discussed this point several times, yes? You remember. And this phenomenon has been discovered, has been expressed by VC. This is a Hungarian great scientist, VC, and also by excellent group of neuroscientists in Karolinska Institute in Stockholm, Fuchse. Okay? Then, neurotransmitters, monoamines, are produced by non-monoaminergic neurons expressing individual complementary enzymes in cooperation. Well, so the same people from Karolinska Institute, they have discovered in the late 80s that there are some neurons which only partly express monoaminergic phenotype, particularly dopaminergic phenotype. Well, so they can express only one between two enzymes of dopamine synthesis. And at least for 20 years, nobody knew what is the functional significance of these neurons. Because individual neurons, they are not capable to synthesize any neurotransmitter. Okay? Well, so, and in our studies in 2000, later, we have proved, we have obtained that these neurons can synthesize dopamine in cooperation. I will explain this phenomenon in more details a little bit later. Okay, and then we have discussed already that neurons are reproduced in adulthood from the progenitor cells. Progenitor is the same as the stem cells. Okay. And finally, neurotransmitters play a role of morphogenetic or of transcriptional factors, in ontogenesis, a role of or transcriptional factors in ontogenesis controlling the development of the target neurons. If anybody could explain me, because I mentioned it already, what is the difference between the action of the same neurotransmitters on the same receptors on the target cells, but during the period of the development of the brain and in adulthood after the full development of the brain? What is the difference? The same chemical signals affect the same receptors on the same cells, but their action is fundamentally different. What is the difference?
Well, did it have something to do with the fact that some neural connections die out through the development of the embryo, or? Well, the connection in an adult is it's more strong because when grew, some connections were deceased. I forget how this process is called, and the connection that stayed are strengthened.
Not really. But look, in adulthood, the neurotransmitter, they provide so-called activating effect on the receptors on the target neurons. What does it mean activating? They provide the action only during the period of association of neurotransmitter with the receptor. after dissociation, this effect disappears. Understand? Huh? For a short time. Huh? Neurotransmitter is associated with receptors and it provides its action, physiological action. After dissociation, there is no the traces of this action. Right? Well, but in ontogenesis, under the development of the brain, if the same neurotransmitters affect the same receptors, this is a long-lasting effect on the development. Well, so it controls not the functional activity of these neurons, but their development, their differentiation. And this effect is long-lasting, so it is maintained for the whole life. Yeah? Understand what is the difference? Not really?
So adulthood is only like the release of the neurotransmitter, short activation, and then stop.
Yes,
And
yes.
in the childhood, it's a long-term effect on the
Yes,
development.
yes. Look, please. Well, neuron, huh? In adulthood, okay? And there is another neuron which provides its action via extrasynatic synapse, okay? Well, so it can activate these neurons by when the neurotransmitters are associated with the receptors for a short period, for seconds. Huh? Well, so it can change this neurotransmitter, can change the functional activity. What does mean the functional activity? On the secretion of the neurotransmitters in these cells, huh? On the generation of nerve impulses in these cells, etc. But this is a very short-term period, huh? Short-term period action. Huh? But during the development, when there is the same interaction of chemical signals on the receptors, this provides a regulation of the development of the neurons. What does it mean? For instance, on the outgrowth of its processes. Huh? On the expression of the genes, for instance, of neuropeptide synthesis. On the expression of the genes of enzymes for monoamine synthesis, etc. And these are the effects which are maintained for the whole life. Yeah?
Does it mean when the neurotransmitter released in the synaptic cleft in childhood, does it mean that it stays in the synaptic cleft longer?
No. It provides also short-term effect. The period of the association with the receptors, it is on the same time as in adulthood.
But the consequences are different.
But the quality of the action is fundamentally different. This is for a short period, only a period of association of receptor, and another one for the whole life. Huh?
That doesn't mean that these effects that are formed in the early childhood and still for the entire life, doesn't mean they're unchangeable or
Yes. And therefore, if there is an impaired metabolism of neurotransmitters or receptors during the period of the development, this is in fact not only a neonatal, as you see, but perinatal period. And after the well, so if there is an impaired metabolism, well, it provokes the development of so-called congenital diseases. Congenital diseases which are maintained for the whole life, and it is practically impossible to correct or to treat these diseases after the development of the brain.
What other examples like ADHD or something?
Well, for one of the examples, this is so-called hypogonadism. Hypogonadism, what does it mean? What's one? This? Well, I will show you. I will show you a little bit later. Look, please. This is the brain, huh? This is a nasal cranium, huh? Nasal, okay. Well, this is a region of the location of the neurons which synthesize gonadotropin-releasing hormones, which control the reproduction in the body. Well, so generation is released into the circulation into the bloodstream, and it provides its action on the gametes, etc., etc. Well, when located in there. But these neurons are generated not in the brain, but they are generated in the nasocranium.
Oh, wow.
Okay? Well, and only after that, they migrate into this brain region. And only in these brain regions, they are capable to control reproduction. But in a disease which is called Kalman syndrome, these neurons are successfully generated here, but they do not migrate to this region of the brain. And in this region, so they function themselves, they function normally in this region. But in this region, they are not capable to provide the regulation of reproduction. And therefore, and this migration, it takes place over embryonic period, or the development of the human.
Before the birth.
So before birth. Well, so that is one of the examples. There are also some examples, but at the level of hypothesis, like autism also it can be, yeah.
Is there any possibility that neurons, not in this disease, but in general, can migrate after the birth, or it's not possible?
You see, but this population of generation neurons,
I mean, in general, the
Yes,
migration.
this is an exceptional population of the neurons, because for most, absolutely majority of the neurons are generated within the wall of the ventricles, of the cerebral ventricles, in the brain. And you remember that ventricular system, Well, so ventricular system of the brain, this is the brain, these are ventricles. Well, so and the epithelium of the wall of the brain ventricles contains stem cells. And stem cells, after the arresting of proliferation, they migrate to the brain regions of the to the place of the final settling, when they provide their specific regulation or their specific function. Okay? Yeah, please.
I will going speak later about methods of migration of cells, because I don't understand how cells can migrate through the brain.
You mean what are the mechanisms? Yeah.
Yeah.
Well, so it is very intriguing question, and it is very intensely studied actually. But something is known. For instance, even in our laboratory, it has been demonstrated that monoamines, serotonin and dopamine, they control migration of the cells, or the neurons, or this is connected, or the outgrowth of actions toward their targets in the brain. Understand? Mm-hmm. Well, so there is a lot of substances, chemical signals, which control the migration of the neurons to the place of their final settling.
Is it really migration, but it's extension maybe or something like this?
The real migration of cells.
In adulthood or in the
No, over the perinatal period.
perinatal.
Before, well, so the neurons, I will tell you a little bit later, but the 99 and practically all the neurons of the brain, they're generated in humans between the 12th and the 16th prenatal fetal weeks. Well, so the period of pregnancy is 39, but they're generated very early. And after that, they slowly migrate to the places of their final settling, and they establish interneural relations. But it's over the perinatal period. And what is important that the regulation of the differentiation of these neurons or morphogenesis of these neurons, it takes place until the end of neonatal life, until the age of 5, 6 years. And this is the most intriguing example that I will give you this example in the next lecture, probably, or after the next lecture. This is a masculinization of the brain, or sexual differentiation of the brain. I mentioned already, yes, that initially the brain is the same in females and males, but over the neonatal period, the male brain in the female brain in males is masculinized, is sexually differentiated to testosterone due to hormones, male hormones, testosterone. Yeah. Well, so that's a really very interesting example.
It's in the neonatal period.
Yes, mostly. Mostly. Yes.
But when we say that neurons can be reproduced, it means that these cells somewhere generated and then go somewhere.
You mean in adulthood, yeah?
Yes, yes.
Yes. Well, so it has been demonstrated, this reproduction of the neurons, or the generation of new neurons, it has been demonstrated in the hippocampus, and it has been also demonstrated that these neurons migrate to the most terrestrial part of the brain, so-called olfactory bulbs. Well, so still nobody knows what is their functional significance. It is believed that this is a mechanism of neuroplasticity. Well, so they can replace the degenerated neurons, but it has not been proved yet. Yet. Okay. Well, okay. Now let's continue. Yeah. Well.
Sorry, last question. You said that the olfactory neurons are generated in the hippocampus and then migrate to the olfactory bulbs in adulthood.
Well, so they generate in the region of the lateral ventricle, in hippocampus, in fact. Okay, and then after all, they migrate to in the reversal direction to put the olfactory bulbs. The very last region, yes,
Yes.
yes, in the adulthood.
So there's a possibility for neurointegration even in a
Exactly. It has been demonstrated. Good. Well, so now we discuss a lot about synaptic plasticity, because this is probably the most important point in neuroplasticity in general. And we discussed, and we will discuss in our seminars several papers on this topic. Well, but what are the mechanisms of synaptic plasticity? Well, so there are specific mechanisms, synaptic, and non-specific. What are the specific? These are the stimulation of neurotransmitter secretion. Well, this is an activation. Well, so if we need to increase the synaptic neurotransmission, that is evident, this secretion, its synthesis and release, should be activated. Then, if we need to increase the intercellular concentration of a neurotransmitter, we should inhibit the enzymes which degrade these neurotransmitters, inactivation of neurotransmitters in the extracellular space by the enzymes of degradation for peptides. How they are called, these enzymes for peptide, this is for monoamines and for peptides. 50 days, a lot of 50 days, specific 50 days. And then an increase of the sensitivity of the target neurons. Do you remember I gave you during our previous lecture an example of so-called upregulation of the expression of receptors by neurotransmitters which are released into the intercellular cleft. Do you remember this example? What is the example?
The example is when a lot of the neurotransmitters, the sensitivity of the receptors decrease.
Yes, yes, exactly. And on the contrary.
Yeah, and otherwise, vice versa.
But I gave you last time, I gave you an example, a concrete example, with a demonstration by positron emission tomography. Do you remember what kind of neurons I have shown you? Dopaminergic neurons. Dopaminergic neurons, because under the increase of dopamine secretion, there is a decrease in the content, in the concentration of receptors for dopamine on the target neurons, and on the contrary. Okay? And therefore, this is when using positron emission tomography, we could specify the speed or the rate or the release of dopamine from presynaptic actions, terminals. Good. Well, and what are the non-specific, unspecific mechanisms? This is stimulation of the growth factors and activation of the antioxidant system. Both of these processes protects the neurons from their death. Well, so these are, in fact, these are neuroprotectors. Growth factors, these are neuroprotectors, and also antioxidant drugs, they're also neuroprotectors. Well, so they protect the neurons from the oxidative stress. Yeah.
By growth factors, do you mean neurotrophies?
Well, so there is a lot of growth factors, and they are peptides. They are peptides, and they, all of them, they provide more or less strong neuroprotective effects, but to a different extent. And therefore, for any neurons, we should select the most efficient growth factors, not all of them, but NDF, for instance, NEF, glial-derived growth factors that are very efficient. So there is a lot of them, okay? But in general, they provide a neuroprotective action. Good, let's go. Well, so, and now return to my favorite neurons, dopaminergic neurons, and what might be the mechanism of their neuroplasticity? What are the indicators of functional activity of dopaminergic neurons? There is in hydroxylate gene expression, synthesis, and activity. Could you remind me? You may ask you to remind me to you. May I ask you how we evaluate the gene expression? What kind of technique we use for that? And for protein synthesis. What technique do we use to specify gene expression? What we measure and what kind of technique we use? Huh? No, this question personal to you. You are very interested in your notebook. But I'm not sure that there is a response. The responses are there, well, on the desk. Could you answer my question? No. I repeat. Well, if anybody could answer my question, please, you wasn't first. Ladies first in general, but...
Okay, so as far as I remember, we used like something fluorescent, like it is in the DNA like sequence that is placed right near the place like which we want to evaluate. Okay, and when like there's an expression of this gene, there's also the expression of this like fluorescent.
Yes, but what is the substrate, the product of the gene expression?
Enzymes.
Not really. Well, so DNA, in this case, DNA serves as a template for synthesis of a certain substance. How it is called, this substance?
RNA.
Messenger RNA. Absolutely, messenger RNA. But how we could detect messenger RNA? With what technique?
Technique?
How it is called? Huh?
Is it staining?
No,
Staining?
no, but there is polymerase chain reaction.
PCR.
PCR. Okay, good. Well, and there is another point. Well, so this is gene expression. This is the first level of regulation, and then the synthesis. Well, so we should detect, we should measure also the protein. The target protein. What is the technique for the study of for measuring proteins, any protein in tissue, in homogeneous tissue? How it is called? Western, the first word is Western, and the second one? Blood, Western blot. Okay, good. Then we should, when showing the functional activity of dopaminergic neurons, we should register dopamine release and uptake. Okay? And what kind of technique can be used to study the monoamine uptake? The monoamine uptake. I'm sure you know this point. No. Okay. Well, listen, do you remember what kind of technique we can use? Yes.
If I remember correctly, it will also stain the precursor of the dopamine that neurons can rotate them.
Right. We should label. We should mark. We should label our precursor dopamine. In fact, dopamine. And then we register on sections or in homogeneity of tissue the specific capture of this dopamine, labeled dopamine, into the cells, into the neurons. Well, good. And what kind of traces, in fact? We can use any trace, fluorescent, but normally we use radio trace, like tree-shaped. Okay, good. Okay, then we should measure dopamine content and turnover, and the monoamine content. What means can be used to measure the monoamine content or their metabolites in the tissue,
The metabolizer monoamines?
Well, sure. We need to measure the content of dopamine or serotonin or any other monoamine in the nervous tissue. What kind of technique we can use for that?
In situ.
Sorry?
In situ.
In
Means in situ.
in homogenate
Oh. We can use.
or in situ. Yes, in cells on sections or in homogenate. Well, in homogenate, this is high performance liquid chromatography. High performance liquid chromatography.
Жидкостных фотографий.
Высокоэффективная жидкостная хроматография.
What about immunoenzymatic?
Immunoenzymatic, yes. We could use also immunoenzymatic assay for monoamine synthesis. But there are two different principles because in this case we use antibodies, specific antibodies to monoamines.
But how does it measure in blood or?
And we can measure anyway. We can measure in homogeneity of the nervous tissue, of the brain tissue. We could measure that in the blood. We could measure it in the cerebrospinal fluid.
Take some
Yeah.
not in situ. It's
Yes,
different.
but no, that is, well, so there are two approaches, in tissue and in cells. Okay, in cells, that you say in situ. This in fact that is in the neurons. What do you mean in situ?
Oh.
Well, so look, in homogeneity, there are two approaches. You are right. This is a monoenzymatic assay, and this is the most frequent high performance liquid chromatography. This is the homogenic. In cells, this is the histofluorescent technique, which is combined with spectroscopy. Well, so we could, on sections, we could differentiate the fluorescent labeled neurons, which produce monoamine, different
But in
monoamines.
slices, not in vivo.
No, but in fact, this is in vivo because you demonstrate, you visualize these monoamines on fixed sections. That's really. But these monoamines, they are contained in survival cells, in survival units. They are not synthesized after they are fixed. I mean that.
But no approaches like to determine
Uh,
minds in live humans, for example.
Yeah,
We have to sample spine, brain, liquid.
I don't remember any technique to study in vivo cells, in survival cells. But probably there was a modification of the histofluorescent technique with the use of black cell accuracy. But it has been developed, this technique, 30 years ago, and it is not used anymore. I don't know why. Yes, sir.
If I understand it correctly, we theoretically can measure the product of metabolism of neurotransmitters in the blood, but it doesn't absolutely correctly corresponds to the level in the brain, right? The level of serotonin metabolic products are not exactly the same, like in the brain.
Yeah, that's true. Well, so first, there is a production of monoamines in the brain. These monoamines diffuse first into the cerebrospinal fluid, into the ventricles, into the cerebral ventricles. Well, so the concentration, because there is no barrier between the nervous tissue and the cerebrospinal fluid, the concentration of monoamines in the cerebrospinal fluid is almost the same as in intercellular clefts in the brain, not in the cells, but in the intercellular clefts. Okay. Well, so these monoamines are not permeable via the blood-brain barrier. But they are produced themselves not only in the brain, but also in the peripheral organs. For instance, the most powerful source of noradrenaline is adrenal glands in the periphery. Well, and also some monoamines can be released from the brain into the general circulation via certain small regions of the brain. How they are called? You remember that I mentioned that there are 5 brain regions, very, very local brain regions, where there is no blood-brain barrier. In fact, there is a blood-brain barrier in the whole brain, but there are small local region with no blood-brain barrier. All these small regions, they are located along the mid-sagittal plane of the brain. You don't remember how they are called? There's a ventricular organs. You remember? But it doesn't matter, but in fact, there are small regions of the brain which are permeable, and therefore monoamines could diffuse from the brain into the blood within this small local regions.
What are the names again of these small regions?
How they're called? Ventricular organs. Sopum, it means that sopum, that is circulation. Ventricular ventricles, cerebral ventricles. Well, so these are regions where the ventricles and this general circulation are connected, are very close to each other. Okay? And they were discovered by an outstanding American Professor Knigge from Rochester. He called them the windows of the brain. The windows of the brain. So-called ventricular organs. Well, okay, now listen. What might be the mechanism of neuroplasticity when coming from this specific mechanism for neurotransmission, dopaminergic neurons? What might be the sorry.
Excuse me, I have a question because I realized you mentioned that we have several techniques to indicate the functional activity of dopaminergic neurons, but at the same time we have non-dopaminergic neurons that have the same function. And what is the technique to like set them apart, like they can analyze them apart from the dopaminergic neurons themselves? Okay.
Well, sure. Well, so there are true dopaminergic neurons with all the functional important molecules you mean, and there are other neurons which also produce dopamine,
Yes.
but these are so-called monoenzymatic neurons.
Yes.
Well, so and what is the difference in their function?
No.
What is the question? Yeah.
Как мы можем при анализе тканей
Дифференцируют дофамин, который продуцируется в одних и в других нейронах.
у которых допамина влияет на да, вот был вопрос, потому что если не посмотрел, в принципе,
A little bit later, I will explain it because this is just the topic of my lecture today. Okay? How we could eliminate dopamine synthesis in these monoenzymatic neurons. Okay? I will answer your question, but a little bit later with a schematic representation. More useful. Well, so mechanism of neuroplasticity is so to maintain the normal intercellular concentration of dopamine and could continue under the functional insufficiency of dopaminergic neurons. Well, so this is an increased tyrosine hydroxylase gene expression, synthesis and activity. Could you remind me again what we measured when studying gene expression? What substance do we measure when studying gene expression?
mRNA.
Messenger.
mRNA. Okay, good. Well, then they increased dopamine release and decreased dopamine uptake, increased dopamine content and dopamine turnover, and increased dopamine synthesis by non-dopaminergic neurons. Well, so this is a compensatory reaction. I will explain the functional significance of this synthesis in non-dopaminergic neurons a little bit later. Well, so just I'm talking actually to this story with non-dopaminergic neurons. Well, so I have shown you that I told you already that these people from Karolinska, they succeeded to demonstrate monoenzymatic neurons, but with no idea what might be their functional significance. And when we studied the arcuate nucleus, hypothalamic arcuate nucleus, many years ago at the beginning of 2000 in my laboratory, and it is known that arcuate nucleus, it contains a lot of dopaminergic neurons. This is a dogma, that was a dogma until this theory. A lot of dopaminergic neurons which provide dopamine, which provide an inhibitory control of dopamine synthesis, of prolactin secretion by adenohypophysis. Well, so dopamine, this is arcuate nucleus, this is the third vertical, this is the median eminence, and this is arcuate nucleus. That's it. Okay? Well, so, and the neurotransmitters, and dopamine particularly, is released into the hypophyseal portal circulation. It reaches the adenohypophysis and it provides an inhibitory control of prolactin secretion via D2 receptors, dopamine receptors, okay? But when we started the development of the arcuate nucleus, we met some unexplained data. First, At the end of fetal life in the rats, look please, these are green neurons which express only tyrosine hydroxylase. They do not express the second enzyme. Red, these neurons express only decarboxylase. They do not express the first enzyme, the second enzyme. Huh? And only yellow, these are very few neurons which express both enzymes. And they can synthesize dopamine, huh? But these are really few cells which cannot synthesize a lot of dopamine, which might be enough to provide an inhibitory control of prolactin secretion. But at the same time, there was an inhibitory control of prolactin secretion. The first question that we made, that in this region, despite almost absent of dopaminergic neurons, there is a huge synthesis of dopamine. But if there are only monoenzymatic neurons that we expressed our idea that this synthesis is provided by two neurons in cooperation. Well, so A-tyrosine is converted in tyrosine hydroxylase containing neurons into a dopa, an immediate percussion of dopamine, and the dopa is captured by neurons which contain only aromatic amino acid decarboxylase for dopamine synthesis. And a dopa should be captured. One moment. By a transporter. Well, so for a dopa, there is a membrane transporter of a dopa, like for dopamine, huh? But in contrast to dopamine transporter, this transporter is not specific. It is also available for large neutral amino acids, like methamphetamine, like tyrosine, etc., etc. Okay. Well, so it's not specific, not only for dopa. Yeah, question, please.
My question is this proportion 99 to 1 is fair only for this brain area, or is it some kind of a general proportion? Listen. So even in adulthood, this is a proportion for the average, huh? At the end of fetal life. Huh? Mm-hmm. But in adulthood, there is many more dopaminergic neurons, about 50%. They're complete, the dopaminergic.
Yes, exactly. Well, so there is a little bit, almost 50% of dopaminergic neurons and 50% of monoenzymatic.
In adulthood.
In adulthood.
And this proportion is fair for.
And this is the case for the late fetuses.
Late fetuses.
Just before birth.
Okay.
Well, so I repeat once again that there is almost no dopaminergic neurons, but there is also a control of prolactin secretion by dopamine. It means that this region, despite it does not contain dopaminergic neurons, it produces a lot of dopamine. The amount which is sufficient to provide dopaminergic control of prolactin secretion.
And how does this proportion changes from 99 to 1 to 50 to 50 in adulthood through the life? Does it mean that the new neurons are generated, or they change their function?
Yeah.
How come?
Yeah, good question. good question, because in fact it's not yet solved, this issue. But I think that mostly to co-expression the second enzyme in some monoenzymatic neurons. Yeah, I do not believe that there is a lot of it's impossible to be generated a lot of neurons out of that. Huh? Okay, even in the late fetuses. But I think that some neurons, they begin to monoenzymatic co-express another complementary enzyme of dopamine synthesis. Okay, I think this is theoretically this is the only way, but still we have no really direct proofs. Look, okay, well, so how we demonstrate, how do we prove that these neurons synthesize dopamine in cooperation? Yeah, and this is an answer, in fact, to a question. Well, so look, well, so if we take the whole region in fetuses or even in adulthood, we know in advance that there is a lot of, in adulthood, 50%, a lot of dopaminergic neurons with both enzymes, and we know that there is a certain population, also 50% of neurons, which express only individual enzymes of monoamine synthesis. How to prove, and the idea was at that time, how to prove that these neurons can synthesize dopamine and calculation. Do you have an idea how to do it?
No. The population of those neurons.
Well, so look, what we have done, and that was in 90s, in the very beginning of, in 90s, I think. And at that time, we had nothing to do in Russia because there were no salary, there were no materials, there was nothing. So we could not imagine that we will be able to use some very sophisticated approaches, expensive approaches, etc. What we have done, we made slices of this region. Slices, these are freshly prepared sections, big sections of the archaeopteryx. And we incubate them in the control, in the buffer, okay? And in the experiment, by adding, by administering a high concentration of A-tyrosine. Because A-tyrosine, it has the same, it also transported, it also transferred via the same transporter as a go. Well, so we realized actually the competition between A-tyrosine and A-dopa. And we have used a much higher concentration of A-tyrosine. And therefore there was no chance for A-dopa to enter these cells.
Because the same transporter is.
It's available for A-tyrosine and for some other
Okay.
amino acids. Well, but if the concentration is significantly high, the competitiveness of tyrosine was significantly higher than for A-dopa. The A-dopa, in this condition, A-dopa could not enter these decarboxylase cells. And therefore, in the whole slices in the experiment, we detected a loss, a decrease of dopamine synthesis.
And what happens with tyrosine in the neurons of aromatic amino acid?
Well,
When it enters, so what happens with tyrosine? In the
with tyrosine,
Yeah.
but tyrosine is used for not only for dopamine synthesis, but it is used for many other proteins. But that was not the topic of our study. But there is no in these cells, A-tyrosine could not be involved in dopamine synthesis,
Yeah.
which should. Well,
But what happens?
what?
Is it like accumulated or really compact? Or
You know, I don't think that it was accumulated. We didn't study it. I don't think that it was accumulated. It was captured, it was released, and it was used for the synthesis of other proteins, like constructive, so-called constructive proteins. But we incubated only for 2 hours, as far as for 1-2 hours. That's not a long period. Well, so that and in this case, what was the and we made these for slices of the archaeopteryx and for slices of the substantia nigra, which contains only dopaminergic neurons as a control. You understand? Well, so we took 2 regions. One region which contains 50-50 dopaminergic neurons and monoamine, and another tissue, substantia nigra, which contains only dopaminergic neurons. Okay? Well, and after these incubations, we can demonstrate that in the substantia nigra, having a pure population of dopaminergic neurons, the dopamine synthesis was highly increased under the incubation with A-tyrosine, because A-tyrosine, or dopaminergic neurons, is a precursor for dopamine synthesis. Well, it is very natural, yeah? Well, so we administered the substrate and we increased the synthesis of the functional protein. But on the contrary, in the acute nucleus, there was a great decrease in dopamine synthesis. Well, and it is evident that this delta, the difference, is the dopamine synthesis, cooperative, so-called cooperative dopamine synthesis. Well, so I mentioned already that these experiments, they were made in the late 90s, I think. Probably I mentioned already because I was very proud of this for this way. Well, so I participated at the International Congress on Medical Alliance in Kyoto. I don't remember in 96, 97, something like this. That was a great congress. Well, and I reported this data, and my colleagues, professors from my country, they were highly excited by these results, in particular by the scheme of the experiment. And some of them, they approached me after, during the break, and he told me that, Michael, now I know exactly that Russian people, they do not have good facilities to make research because they have excellent brains. I was proud of them, not for me, but really for Russian people, because they always are obliged to use their brain sometimes instead of excellent facilities which are available in Western countries. Well, now let's go. Well, and there is a schematic representation. Well, so finally, we came to two conclusions. The dopamine is synthesized by non-dopaminergic neurons expressing individual complementary enzymes of dopamine synthesis in coagulation, and coagulative synthesis is a compensatory reaction under a failure of dopaminergic neurons. For instance, in neurodegenerative diseases, under the functional insufficiency of dopaminergic neurons. I will show you the evidence on the next slide. Well, so this is a schematic representation, in fact. So A-tyrosine is converted in dopaminergic neurons, first to L-dopa and then to dopamine. But in monoenzymatic neurons, it is converted first into L-dopa, and then L-dopa is captured by decarboxylase containing neurons, and it's converted to dopamine. This is schematic representation. What is interesting? That L-dopa is synthesized in tyrosine hydroxylase containing neurons. Okay? But L-dopa, it can be captured by different neurons for further synthesis. For instance, it can be captured to decarboxylase cell, like this. This is a cooperative synthesis, which is demonstrated here. L-dopa can be captured to noradrenergic neurons, which contain both and it can be captured to serotoninergic neurons. Well, so if it is captured to serotoninergic neurons, they begin to produce dopamine instead of serotonin. Interesting, yeah?
Can it be vice versa?
Yes, well, so,
Can dopaminergic neurons produce serotonin?
serotonin. Well, on the contrary, no, it was not shown.
Only one way.
Yes, only one way. Well, so I repeat one again, that if we will introduce this dopamine, if it is captured to serotoninergic neurons, which contain tryptophan hydroxylase, not tyrosine hydroxylase, but tryptophan hydroxylase. This is a specific enzyme for serotonin synthesis, and decarboxylase, which is no specific enzyme for monoamine synthesis. Well, so if it is captured here, it can be converted by decarboxylase to dopamine. And this has been demonstrated, but not On the contrary.
But if tryptophan is captured by dopaminergic neurons, logically it can be the same way. That the
If a dope is captured too,
no, not in tryptophan, the precursor for serotonin is captured by the complete dopaminergic neurons, and then the decarboxylase transferred to serotonin.
you know, everything what was said about cooperative synthesis, that has been proved for dopaminergic neurons.
Oh,
But most probably that might be the case for serotonin, for neurons which express individual enzymes of serotonin synthesis. They have been demonstrated, but decades ago, and nobody took care about these neurons. But we began to study them. First, we're trying actually to make mapping all through the brain. If this is a great population of monoenzymatic, for instance, which express, for instance, only tryptophan, hydroxyl, and so on. But it is most probable that there is the same cooperative synthesis for serotonin, like for dopamine, but it's not proven. Well, so in my laboratory, well, so I told you that we try to prove that this cooperative synthesis is very important as a compensatory mechanism in neurodegenerative diseases. Well, so we have reproduced Parkinson's disease in animals, in rodents, in mice, yes, in mice. Well, so look, please, I remind you again that under Parkinson's disease, there is degeneration of dopaminergic neurons in the substantia nigra, and differentiation dopaminergic, and a loss of dopaminergic neurons, which innervate striatum. And these neurons are responsible for motor function, for the regulation of motor function. Well, so in Parkinson's disease, there is no this way for regulation. Okay. Well, and in the striatum, in normal mice, in normal humans, there is almost no dopaminergic neurons. So, but look, what we have demonstrated. The striatum of normal animals containing monoenzymatic neurons expressing individual enzymes. That even in the striatum there are monoenzymatic neurons. Then we have demonstrated that monoenzymatic neurons expressing tyrosine hydroxylase or aromatic amino acid synthesize dopamine and copulation, but only under loss of dopaminergic neurons, not in normal. In the archaeoglyphs, there is a cooperative synthesis even in normal animals and normal mammals. But in the striatum, only under a loss of dopaminergic neurons and differentiation. Dopamine synthesis by striatal monoenzymatic neurons is enhanced as dopaminergic neurons degenerate. Please look here. This is a control with a high 100% level of dopaminergic neurons. This is dopamine synthesis. And this is dopamine synthesis at different level of differentiation of the striatum. At the lower level, there is a dopamine synthesis by monoenzymatic neurons and 70% by dopaminergic, but not neurons, but the axons, which are coming here. Okay, and under the progressive degeneration of dopaminergic neurons in the substantia nigra, there is an increase of the proportion of monoenzymatic of dopamine synthesis by monoenzymatic neurons when comparing with benzymatic neurons. Okay, well, so this is a compensatory reaction. Dopamine synthesis by monoenzymatic neurons is an efficient mechanism of neuroplasticity in Parkinson's disease. What is the most interesting point in these experiments and what we are doing actually? Because look, we know that, and I will repeat it again during our next lectures, that Parkinson's disease is developed because of degeneration of these dopaminergic neurons in the substantia nigra and degeneration of the striatum. And this is a progressive degeneration of the neurons. For 20, 30 years, there is a degeneration of these neurons, but without any manifestation, external manifestation in motor behavior in people and in animals. Huh? And only after 30 years in humans, there are motor symptoms which manifest. Okay? Yeah, proceed.
I have a question according to the information you previously said about the treatment of Parkinson's disease. You said that we can prescribe L-dopa and dopamine for the patient, but this treatment lasts for 2 or 3 years, and then it's
It becomes toxic.
and yes, and at the same time you mentioned that there might be a situation when the transporters, this type 2 transporters won't be enough for the those
The first.
molecules,
Hmm.
and but couldn't we also treat the person with those transporters as well, or something to not only to use this dopa and dopamine, but also introduce with transporters, so it won't be that toxic and lasts longer.
Well, I don't see already that this might be the best idea, but in fact, instead of L-dopa, the people, physicians, they use so-called agonists of dopamine, also substances which can react with dopamine receptors. Well, but dopamine is not permeable via the blood-brain barrier. But these agonists, which are synthesized artificially, they're permeable. And therefore, we can use this artificial dopamine, which affect the dopamine receptors.
Okay.
Well, so there are 2 ways: L-dopa or dopamine agonists. Okay. Well, but I return to this one. Well, so and if the disease is developed for 20, 30 years without any manifestation of motor impairment, it means there should be compensatory somehow compensatory mechanism. And all the people, they consider as compensatory mechanism the functional activation of these dopaminergic neurons. Well, so which remain survive, okay, which remain survive. But in our laboratory action, after discovering this mechanism of cooperative synthesis of dopamine in the striatum, we can see that this probably even much more useful compensatory mechanism for the nivelating of the deficiency of dopamine under the degeneration of these dopaminergic neurons. And therefore, we are studying actually the molecular mechanism of this cooperative synthesis in the striatum, because striatum itself is involved into the regulation of motor function. Okay? Even striatum. And we have demonstrated that in addition to the neurons of the substantia nigra, there is another source of dopamine. These are striatal neurons, monoenzymatic neurons. And we can see that this might be much more important compensatory mechanism. And we are studying the molecular mechanism for regulation of this cooperative synthesis, and we believe that this mechanism should be used, the knowledge of it should be used to develop new technique for the treatment of patients. So this is one of the ways to follow the development. Well, I think, yeah, please, you're welcome.
So, to understand correctly, that the generation of the dopaminergic neurons continues to happen in this rate, and that's why this compensatory mechanism over time stops.
I repeat again that in Parkinson's disease, there is a progressive degeneration with time, slow degeneration of dopaminergic neurons which are located in the substantia nigra. But dopamine is transferred via axons, which is synthesized here, into the striatum. And into the in the striatum, dopamine provides its neurotransmitter function for the regulation of motor behavior. Okay? But we have demonstrated that this dopamine, which is synthesized in these neurons, this is not the only source of dopamine in the striatum. That in the striatum, there are monoenzymatic neurons, interstriatal monoenzymatic neurons, which can synthesize dopamine first, and which increase dopamine synthesis under degeneration of dopaminergic neurons of the substantia nigra. Therefore, we consider that this cooperative synthesis of dopamine in the striatum is an important, most important probably, compensatory mechanism, and we will use this to develop a new therapy for dopamine for Parkinson's disease.
My question was more about, you say that this mechanism starts to happen even while we can observe the symptoms, right?
Wait, what?
When the symptoms are not visible,
Yeah.
right? So why over time the symptoms become visible? It means that this compensatory mechanisms after some time can compensate.
Yeah. Yes, because this motor dysfunction, it first manifests under degeneration of 50% of dopaminergic neurons. But then there is a the degeneration continues. And therefore the compensatory mechanism, this synthesis in the stratum, in the animals, it can improve the situation. It could compensate the dopamine synthesis under a certain level of degeneration of these dopaminergic neurons and of a loss of dopamine which is synthesized in these neurons, okay? But no more. But if we will be able to control this compensatory mechanism, probably we will be able to improve its efficiency.
So
On the first, this is the first point. On the other point, I told you that we're trying actually to study so-called preclinical study of the development of Parkinson's disease before the manifestation of motor symptoms. And at this stage, there is a loss of dopamine less than 50%, for instance, 20%. And in this case, it's much more important to prolong the survival of these neurons. Well, so this neuroprotection, and we're using it. Okay, you got a question? No?
Yeah, but I guess I will ask it in the break.
Yeah.
If it's possible.
Okay,
Not so.
Okay, you have a break.